Hey guys, we are back and we are going to start talking about the specimen. So, instructions for properly procuring, storing, and handling and transporting laboratory specimens can be obtained from each independent laboratory. So, if a provider gives you an order to get a certain specimen and you've never done it before, you can always call the lab and they will give you directions on everything. So, obtaining the specimen in the proper manner and using the right equipment will ensure that a high quality, accurate specimen is submitted to the lab. So that's so important. Always ask questions if you are unsure of what to do or how to procure a specimen from a patient. There are some guidelines to follow. You obviously want to check your provider's orders and make sure that you have the right patient, of course. Refer to the lab instruction manual or consult the lab, as I just mentioned. You want to instruct the patient on any dietary restrictions. So, for example, oftentimes if you have an annual physical exam, they want to do a blood test on you, and they will tell you nothing to eat or drink from midnight on until you get your blood sample taken. So make sure that the patient has followed that. Instruct the patient to ingest special food or take substances if required. Sometimes you may need to take a specific pill or something that they give you. For instance, when you're pregnant, they give you a very high glucose drink, and then you come into the office and then they check your blood sugar. So things like that. Select the appropriate container. It's very important, especially for certain blood tubes. They have different additives in the tubes, which we'll talk more about later in the semester. But different additives. It's not just any old tube will do. There are specific ones for specific tests. You want to label the specimen correctly. And we always label the container, not the lid, because lids can be taken off, right? And then, which lid went with which specimen. Label the container, not the lid. You want to obtain the specimen or instruct the patient to provide specimen according to instructions. So be very clear if the patient is getting the specimen for you, for instance, a urine specimen, it's important to tell them exactly how to do so. Of course, follow bloodborne pathogen guidelines. Place any specimens to be sent to an outside lab in an approved bag, and always document what specimen you obtained, where it's going, and all of that. Because remember, if it's not documented, it's not done. All right. So moving on to some of our tests that get ran. Remember our different departments. Hematology is a department that they're looking at the formed elements of blood. The study of blood cells and coagulation in both normal and diseased states. There are two different main components of the blood. There is plasma and the cells. So the plasma is the liquid portion, and then the cells are the formed portions that are floating around. The cellular components, so those cells, are made up of erythrocytes, which are red blood cells, leukocytes, white blood cells, and thrombocytes, which are platelets. And hopefully this is a reminder from some of your science courses you've already had. Now within hematology, there are certain tests that are performed like hemoglobin, hematocrit, white blood cell count, a differential white blood cell count, red blood cell indices, platelet count, erythrocyte sedimentation rate, or prothrombin time. And we're going to go over some of these next. So within hematology, hemoglobin is a very common test to be ran, and hemoglobin is the major component of the red blood cell, and it serves to transport oxygen and carbon dioxide through the body. So very important, because we need oxygen to be for all of our organs to function. Hemoglobin carries about 95% of the oxygen that the body cells use, and it carries about 27% of the carbon dioxide. So super, super important for our body. There are different forms of hemoglobin. Hemoglobin A is the most common, but just note that there are different forms of it. The most frequent hemoglobin disease seen in the clinical setting is anemia. Iron deficiency is also the most common type. So this is caused by not enough iron in the diet most of the time. And so eating green leafy vegetables and meats that have iron, very important to make sure that our iron levels are up and decrease the likelihood that we have iron deficiency anemia. Note here that on these pages I do give you the general range for our lab tests. So for hemoglobin, for a female, it's going to be 12 to 16 grams per deciliter. For males, it's going to be 13 to 18 grams per deciliter. I do not need you to memorize all of these, but it's important to kind of just see what those ranges are and notice that they differ a little bit between men and women. So hemoglobin can be tested within the physician office laboratory using a CLIA waived POCT device. And these devices are infrared analyzers that measure the density of the hemoglobin pigment by light refraction. Sounds way high tech. But it's a quick method, and it only uses a small drop of blood, and it gives you immediate results, and it tells you that hemoglobin level. So you'll see here that hemoglobin specimen from that drop of blood that is applied to the device is 13.6, which is great. There is a video for hemoglobin analysis if you want to watch that and to learn a little bit more, because you may be using it within your clinic. Hematocrit. So along with hemoglobin, you often look at the hematocrit level of a blood sample, and this is the ratio of the volume of packed red blood cells to that of the whole blood specimen. So here you will see we had a whole blood specimen. It was placed into a centrifuge, which is this, and those blood components separate. So you'll see here this yellow part is the plasma, and remember that's the liquid portion of the blood. We have a little buffy coat, which is kind of the separator, and then we have our packed red blood cells. And for females, we want 36 to 46% for our hematocrit level. So if you notice, that looks about right because we are measuring the ratio for the packed red blood cells. So the packed red blood cells should be 36 to 46% of the entire specimen. As I mentioned, the components are separated by spinning the specimen in a centrifuge at high speeds. And it's measured on a scale. So you place this, once it's been separated, onto a scale, and it gives you that percentage number. All right, another test within hematology is our leukocytes, our white blood cells. White blood cells help protect us from infection. There are 5 different types of white blood cells, each one providing a specialized way of protection. These methods include phagocytosis, detoxification, inflammation, and immune response, and they help fight against the bad guys in our body. A differential count for our leukocytes tells us about each type of white blood cell. So you can get a normal, just a leukocyte reading, or you can get a breakdown of which type of leukocytes you have in your body at what percentage. You can see that here, normal values of a differential leukocyte count, and then just your normal leukocyte count here. Just to mention, leukocytes, testing those is not CLIA waived, so you will not be testing those within your own physician office laboratory. All right, red blood cells or our erythrocytes. Remember that our red blood cells are responsible for carrying blood. Remember, hemoglobin and hematocrit are a part of that. An immature erythrocyte, a red blood cell, is called a reticulocyte. If there's an increase in the number of reticulocytes, it's an indicator that the body needs more because it's making new baby blood cells, red blood cells. Erythrocytes can be various sizes, and they're called normocytic, macrocytic, meaning big, or microcytic, meaning small. They are mostly round or slightly oval in shape, which you can see here. And both white blood cells and red blood cells can change in reaction to disease. And you'll see here the normal ranges for red blood cells. All right, platelets or thrombocytes. They are involved in the clotting of our blood for coagulation. The normal number of platelets is 150,000 to 450,000 per microliter of blood. If it's above 450,000, it's said that the patient has thrombocytosis, and these high platelet counts can be caused from cancer, anemias, and other conditions. If it's below 150,000, the patient is said to have thrombocytopenia, and this is caused, or these low counts can be caused by leukemia or certain types of drugs. Guys, I took care of a patient recently, and their platelet count was 7,000. Extremely low, very life-threatening, and it was caused by a drug. A drug had actually, a prescription drug, had caused this thrombocytopenia and was very dangerous. Luckily, it is treatable, but it was very interesting. All right, erythrocyte indices. These include MCVs, MCH, MCHC, and these calculations provide information about the sizes of the red blood cells and the hemoglobin content. I don't want you to read too much into this. It's important to have just heard this, but again, you don't need to know all of the details. These readings or levels tell us about different diagnoses and different types of anemias. Then erythrocyte sedimentation rate. This is the measurement or the rate of which red blood cells settle towards the bottom. So they're floating around, but eventually they settle at the bottom of a specimen. In normal blood, erythrocytes tend to remain suspended in the plasma. Like I said, they're floating around. But sometimes there's a phenomenon in which these red blood cells form clusters and they clump up, which causes the rate of sedimentation to increase. There are two traditional methods for performing the erythrocyte sedimentation rate. One is called the Wintrobe method, and these are the ranges that you actually get from that method of testing. And there's also a Westergen method. All right, coagulation studies are INR and ProTime. The method of monitoring coagulation time is called prothrombin time, or PT, or it's more commonly known as ProTime. It's reported in seconds, and it reports how many seconds it takes for the patient's blood to clot, and it's also known as the International Normalized Ratio. Normal blood will clot in 11 to 13 seconds. If you're on an anticoagulation medicine, you'll want a ProTime of 16 to 18 seconds with an INR of 2.0 to 2.6. If the blood clots too soon, then it means the anticoagulation medication is not therapeutic. It's not doing the job that you need it to do. ProTime is a simple CLIA wave test, and there is a video listed here. You'll need a small sample of blood that is processed into a piece of equipment, and it gives you that ProTime reading. Okay, so those were all hematological tests that we just mentioned. I do want you to note here that we have a whole list of blood chemistry tests. There's all type of chemical analysis that you can do on blood, and these are just a few examples here. Just to point out a few, bilirubin tells us about our liver and breakdown of protein in our body. It also, some other ones, potassium. Potassium is really important for muscle contraction and electrical impulses. We have calcium, creatinine, which tells us about our kidney function, all different types of tests that can lead us to different interpretations of disease processes and things like that for the provider to make diagnoses. All right, so moving on from blood samples to urinalysis, just going to barely bring it up here, and then we'll move on in our last video to kind of describe it. But urinalysis is the study of urine to diagnose and follow different courses of diseases. After urine processes through a healthy kidney, it's approximately 96% water and 4% dissolved substances. And I want you guys to take some time here to look over what is normal to be found in urine and what is abnormal. And remember, this may come up on a quiz question.